come to our last speaker. Another talk I'm really looking forward to because this will be held by Alessandra Griffo. She is currently a postdoc in Kerstin Göpfrich lab at the Max Planck Institute for Medical Research in Heidelberg. And they are working on, RNA origami pores. So it's really tailor-made, not DNA pores, but RNA pores. She will tell us why they do that. It's pretty exotic. It's very interesting. So really looking forward to this talk. Yeah.
Okay, so hello everybody. Thank you, Conrad, for the nice introduction. So I started my postdoctoral research last year at the Max Planck Institute in Heidelberg, and today I will talk about my postdoctoral project, which aims to the design and the characterization of RNA origami nanopore. I know the topic might sound quite exotic. We already had the pleasure to see in this section how nanopores can be made by the self-assembly of different materials, like, for example, from the self-assembly of protein pores, which are studied for better understanding biological phenomena and also for sensing, like we have seen. We also saw peptide-based nanopores, which can be used for kill bacteria and so on. But recently, in the last decade, there was a field in continuous expansion, namely the DNA nanopore nanotechnology, which aimed to develop the design of DNA-based nanopores. Why it would be interesting to use DNA-based nanopores? Where they are amazing for de novo applications, because with the technique of the DNA origami, you can easily assemble and pores with precise and tunable dimension, charges, and properties. Our goal, however, is to shift and to move from the design of the DNA nanopores to RNA origami nanopores. Because DNA nanopore technology so far has been amazing for a huge range of applications like sensing, drug delivery, DNA sequencing, and so on. But there has been a limitation. A limitation is that for making DNA nanopores, we know that they do not spontaneously interact with the lipid membrane. They need the chemical modification. So we need to interfere with the structures and very often functionalize the DNA with hydrophobic anchors, namely cholesterol anchors or porphyrins and so on. That's the only way how the DNA pore can assemble and interact with the lipid membrane, insert, and form stable pores. The advantage of RNA origami nanotechnology would be to overcome this limitation, meaning that we, for scaling up the project, we would not encounter high-cost production. Also, RNA is much more biologically relevant. In fact, we can easily transcribe from a DNA template our origami, and the reaction will be simply enzymatically triggered by a T7 polymerase and thermically activated. And we can fold our origami in the desired shape by simply co-transcriptional folding. The goal of the project, therefore, would be to create a RNA-based nanopore which spontaneously interact with lipid membrane, insert, and form pores, which we want to use as biophysical tools. So we are working with a model system with freestanding lipid membrane, black lipid membrane. But our ultimate goal, since the highly biological relevance of RNA, would be then to go for co-transcriptional folding inside synthetic cells and to encode synthetic cells for permeability, for example, and use as very powerful biophysical tool. So in order to realize our goal, to achieve our dream, the first step we need to do is to sit on a computer and to develop, to design our RNA structures. So we need to start from making an arrangement, and we choose structures which features and display 8 RNA duplexes, which are interconnected via 3 dovetails that are base paired, and they arrange around a central unit, forming a nanopore which has a length of 27 nanometers and a diameter around 8 nanometers. These estimations are done via RNA simulation. As I said, we know that RNA can be a much powerful tool because we might have, for example, RNA aptamers which can spontaneously interact with the lipid membrane. In our design, that's actually the first design ever for RNA nanopores able to insert into the membrane. we inserted 8 repetitive motifs, which we called loop out. Those motifs are able to base pair with a complementary sequence of DNA, which carry our binding aptamers of interest that want to lead the nanopores to insert into the membrane. So after we design our RNA structures, we of course had to characterize and to produce our material. So we ran an in vitro transcription, and the best tool that I chose to characterize the transcription of our template was the technique of atomic force microscopy. Because with this technique, we can see not only if the transcription occurred, as you can see from the morphology pictures here, we have simply the template treated with DNA, and here we run the in vitro transcription, but we can also have a look at the structures deeper with high resolution pictures and see if the nanopore was also folded correctly, because we need a folded pore, or being is an open sheet. And as you can see on the pictures on the right, in the majority of the cases, the nanopore folded correctly. And we can see by looking at a cross-section analysis that features a length around 13 nanometers and a height around 8 nanometers, which match with the sizes estimated via RNA simulation. At this point, we designed our nanopore, we transcribed our nanopore. We need to look up the binding anchors that are needed for bringing into contact the nanopore to the lipid membrane. As I mentioned before, in the DNA nanopore nanotechnology, cholesterol anchors have been the leading hydrophobic anchors to be used to let interact the pore with lipid membrane. We have already a showcase of tons of examples where nanopores were stably inserting in lipid membrane, and actually the insertion event were triggered via nanopore current measurement. Therefore, we choose as a control system nanopore design, which is an hybrid where we choose the loop out region to bind to the complementary base pair with cholesterol anchors. However, our goal is to create a totally RNA origami based pore. Therefore, we moved to RNA aptamers, namely biotin aptamers, which accommodate quite nicely the biotin, so establish a good binding with biotin between the nucleotide, those two nucleotides, so between the loop 2 and the helices 2 of the pseudo-knot structure. So this will be our second candidate structures, namely totally RNA origami based pore. Before to start our to test if the pore we produced is membrane active, we needed again to run some control experiments. So first, we wanted to prove the attachment of the biotin aptamers to the RNA pore. And to do that, we again employed the technique of atomic force microscopy. And we could see a difference between the image of the pore itself and the pore which contains the biotin aptamers. In fact, the structure below appeared to be much more bulky if you look at the set height profile. And we could also really identify the biotin aptamers all bound around the nanopore. And secondly, also in our buffer condition, we wanted to test that the building block of biotin aptamer was actually binding to lipid membrane. And to do this, we ran some assay with confocal microscopy, some binding assay. And as positive, as negative control, we generated liposomes, namely G and unicellular vesicles, and we injected the fluorophore-tagged DNA, which was not supposed to bind to the lipid membrane. In fact, we observed only a red background. And after that, we inject our biotin aptamers, which can bind again with the same technique of base pairing of complementary DNA strands, our fluorophore, and we observed via confocal microscopy a ring around this black hole, which is the not stained G and unicellular vesicles, meaning that the biotin was binding quite nicely. At this point, so just to have a recap, we had our design, we had our nanopore, we transcribed the nanopore, we verified the attachment of the binding anchors to the nanopore. We want to test if the nanopore is membrane active. And that's also the reason why I'm giving the talk here, because we used the Orbit Mini device. So we scanned with the lipid bubble technique lipid membranes over make a chip displaying a circular cavity around 100 micrometers and test first the stability of the membrane, since we have a mixture of the PHPC and biotinylated lipids, since we need the biotin to bring together the biotin aptamers. And we test, of course, buffer conditions, and also we looked at the current graphs of the single building block, namely the biotin aptamers and the RNA origami pore. And we observed that in both cases there was not activity, which was very nice because the pore itself should not interact at all with the membrane. The biotin aptamer should bind to the membrane, but should not lead to any insertion. So what we observed was sometimes a tiny disturbance, but never a real insertion event. At this point, we wanted to test the two pore candidates, the hybrid DNA RNA pore, for which we had higher expectations since we know that the cholesterol anchor inserts quite nicely into the lipid membrane, and the totally RNA origami pore. For the the DNA RNA hybrid pore, we observed some transient insertion events, but very often we observed the disturbance of the membrane after insertion. We also drew a statistics of the current events that we recorded, and we obtained a conductance around 0.8 nanosiemens. For our totally RNA origami pore, indeed, we also repeated the same measurement, and we very nicely observed the typical current traces. These are actually classical fingerprints for DNA nanopore insertion, proving that there was a nice insertion of the RNA pore when the biotin aptamers are used as binding anchors. We also, in this case, detected the statistics for the current dump recorded, and we got a value of around 0.9 nanosiemens in conductance. So far, I also have to say that the rate of success of this experiment was not very high because the nanopores do not insert so often, and we are still working to improve that, increasing the concentration, because also for RNA transcription, the yield was not so high so far for this huge structure. I think that we had a pore which is almost 2 kilobases. And so the conclusions so far for the project are that we have been able to design and produce and characterize the first RNA nanopore, which is able to interact with the membrane, to be membrane active. And we have some perspectives. So what we aim to do now is to look, once we will have increased the statistics of insertion event for the totally RNA origami pore, we would like to tune the voltage to see how the pore respond in function of the voltage applied, because we know that drawing the IV records, we can get information about the nanopore dynamics and also the conformational changes or getting, which we know that can be voltage dependent. With these findings, I come to the conclusion. I would like to thank my group, in particular Casting Office, for the support, and Eric Poppleton that helped a lot with the design. And of course, I thank you all for the attention.
Thank you so much, Alessandra. Are there any questions from the audience or RNA pores? Yes.
Thank you for the interesting talk, actually. My question is, how stable are the RNAs in your solution? Because they're underlying hydrolysis usually.
Yes, yes. We also wondered at the very beginning if we would have had some issue in terms of RNA stability, but once we form the origami structure itself is quite stable. Actually, it can last till weeks without being degraded. So that's great. But once the structure is properly folded, okay. So of course we have to be careful to environment, to hydrolysis and so on. We always keep pH below 8. Yeah, we take all the precautions needed.
Keep out RNA.
Further questions? Anything? So, I mean, you're just getting started with doing IV traces. That would be very interesting to see if you can observe any gating.
Yes.
Analogous to what you see for
yes,
biological
we are also
if it's reversible.
Yeah, we're also very curious to see if we would observe rectification in the IV, because RNA is negatively charged, so it won't respond symmetrically to the IV record.
All right, great. Let's thank Alessandra again.